Imagine trying to bake like a very specific layer of a multi-tiered cake.
Yeah.
You have your recipe, you carefully measure out the flour and the sugar, you set the oven perfectly.
Right.
But then when you pull the pan out, half of that layer has magically turned into an entirely different dessert.
Like a completely different pastry.
Yeah, exactly. Like you wanted a vanilla sponge and you ended up with, I don't know, half vanilla sponge, half cherry pie.
Which, I mean, in a culinary setting, that's just a total disaster. You throw it out.
Right.
But in the world of neuroscience, observing that exact kind of spontaneous, totally unexpected transformation recently led to a massive breakthrough, like a real paradigm shift in how we understand human brain development.
And that accidental cherry pie is exactly where we are starting this deep dive today. We are looking at this landmark paper. It was published in Nature Neuroscience back in June of 2026.
Yeah, it's a huge paper.
It really is. So scientists were cultivating these things called organoids, right? Basically growing 3D models of the human midbrain in a lab. But when they looked closely at the resulting tissue, they realized that over half of the cells they created, well, they weren't midbrain cells at all.
No, they're from the hindbrain. Completely different region.
Completely different. So our goal for this deep dive is to basically decode every single piece of this complex biological puzzle. We're going to break down the technology they used to track those cellular decisions, how they mapped out the physical neighborhoods of these mini brains, and ultimately how they're engineering custom brain tissue to study diseases like Parkinson's and autism.
Because the researchers didn't just toss the experiment when it went wrong. They wanted to know exactly why those standard protocols were producing the wrong brain region. And, more importantly, how they could map the genetic decisions those cells were making in real time so they could eventually just take control of the whole process.
Right. And if you're listening and thinking, wow, that sounds like a lot of heavy terminology, don't worry. We are going to break down every single mechanism here.
Yeah.
But to understand how they uncovered this midbrain, hindbrain mix-up, we have to look at how they tracked the development of these organoids over time, right?
Exactly. They didn't just look at the final product on day 120 and say, oh, well, that's weird. They monitored a staggering 104,452 individual cells.
Over 100,000 cells. That's just wow.
Over 120 days. And tracking that kind of volume, it requires a dual approach, which they achieved using a single-cell multi-omic atlas.
Multi-omic, meaning multiple layers of data.
Right. They collected 2 distinct layers of data simultaneously from the exact same cells. So the first layer is the transcriptome, utilizing RNA sequencing.
Okay, RNA-seq.
Yeah. And this captures the active output of the cell, like which genes are actively firing or being transcribed into RNA at any given moment.
So that basically tells you what the cell is currently doing right now.
Exactly. But the second layer, this is where it gets really interesting. It looks at the physical structure of the DNA itself. They use something called ATAC-seq to map accessible chromatin.
Okay, wait. Accessible chromatin. Break that down for me.
Sure. So if you think about the DNA inside a cell nucleus, it's about 2 meters long if you literally stretch it out.
2 meters. In a single microscopic cell.
Yeah, it's wild. So to fit inside that microscopic space, it has to be tightly spooled around these proteins called histones. And when DNA is spooled up like that, it's silenced. The cellular machinery just can't reach it.
Oh, I see.
So ATAC-seq uses a hyperactive enzyme to tag only the unspooled exposed regions of the genome. It basically shows us the open chromatin, the stuff the cell can actually read.
Okay, so think of the cell as a massive manufacturing plant, right?
Oh, wow.
The RNA sequencing the transcriptome, that's like the clipboard on the factory floor telling us exactly what the workers are building on the assembly line today.
I like that, yeah.
And then the ATAC-seq, the accessible chromatin. That tells us which filing cabinets of blueprints are currently unlocked and available up in the manager's office.
That is a perfect analogy. And using that ATAC-seq data, they found 243,296 specific genomic regions that were open and accessible. And those regions contain the regulatory elements that dictate what a cell is going to become in the future.
But wait, if we already have the clipboard, the RNA telling us exactly what the workers are building right now, why do we care which filing cabinets are unlocked? I mean, doesn't the RNA just give us the definitive answer of what kind of cell we're looking at?
It gives you the present reality, sure. But the open chromatin gives you the latent potential.
Potential.
Right. A filing cabinet might be unlocked with the blueprint literally sitting right on the desk, even if the workers aren't using it yet. By mapping the accessible chromatin, scientists could identify these hidden regulatory networks. They could literally see the instructions for becoming a hindbrain cell sitting there, fully accessible, long before the cell actually started manufacturing hindbrain proteins.
Wow. So they are looking at the cell's capacity for change, not just its current state.
Exactly.
So for everyone listening, let's do a quick recap. Basically, organoids are these 3D lab-grown brain models. And getting the midbrain versus hindbrain right is crucial for studying motor and sensory functions. And this multi-omic tech reads both the active instructions, the RNA, and the open blueprints, the chromatin.
Right.
So think about your own daily workflow for a second. If someone only looked at your active output, like what you're typing right now, versus all the reference tabs you have open in your browser, what would they miss about your actual process?
They'd probably miss where you're heading next.
Exactly. So if these hidden blueprints reveal a cell's potential, what did the researchers actually find when they dug into those standard midbrain organoids? You know, the ones that turned into the accidental cherry pie.
Well, they scrutinized two widely used existing protocols for growing midbrain organoids. Protocol 1 bathes the cells in a continuous chemical signal using molecules known as CHIR, SHH, and FGF8. And this approach is generally considered, successful for generating dopaminergic neurons, specifically A9-like neurons, which are the exact cells that degrade in Parkinson's disease.
Right, which is why everyone wants to study them.
Exactly. Now, Protocol 2 applies signals sequentially in distinct stages, and that one is known for producing glutamatergic neurons.
And both of these protocols were already designed, published, and basically used everywhere by the scientific community, right? Everyone just assumed they were creating relatively pure midbrain tissue.
Yeah, that was the assumption. But when the researchers applied their multi-omic tracking to these organoids, they found that a massive 57% of the generated cells were actually hindbrain cells.
By over half.
Over half, 57%. They were making things like glycinergic neurons and Purkinje cells.
Why? Like, how does that even happen if you're following the recipe?
Because the chemical signaling gradients in a lab dish are incredibly blunt. If you compare them to the precise microscopic delivery systems of a developing human embryo, it's like a fire hose versus a scalpel. Because of that bluntness, the cells were getting mixed signals. They were accessing those open hindbrain blueprints we talked about and differentiating into the wrong tissue entirely.
So if the chemical signals are that messy, and the resulting tissue is this 50-50 mix of midbrain and hindbrain, it sounds like it would just be a chaotic jumble of cells.
Yeah.
I mean, how do you even begin to study the structure of an organoid if it's just a disorganized blob?
Well, that's the thing. They weren't actually in a random jumble. But to prove that, the researchers had to map the organoid physically.
Okay.
And that requires moving beyond standard RNA sequencing. Usually when you sequence RNA, you have to dissolve the tissue, which completely destroys the physical architecture. You lose the map.
Right, you blend it all up into a smoothie.
Exactly. So to solve this, they used a spatial transcriptomics tool called MERScope.
Okay, MERScope, which stands for multiplexed error-robust fluorescence in situ hybridization, right?
Yeah, it is quite the mouthful. But the key word there is fluorescence. MERScope uses combinatorial labeling. Basically, it assigns a unique fluorescent barcode to different messenger RNA transcripts.
Oh, wow.
So this allows scientists to take a physical, intact slice of the organoid and map the exact physical location of hundreds of different genes firing simultaneously. It acts as a highly detailed GPS map.
So they're literally lighting up the specific genetic activity of the cells, like a city grid at night.
That's exactly it. And that GPS map revealed that the organoid was remarkably structured. Despite the totally chaotic chemical signals they were bathed in, the cells had communicated with each other and self-organized into highly distinct, segregated geographical neighborhoods.
That is wild. The paper actually maps out several of these neighborhoods, right? But two really stand out to me. The areas expressing the genes SHH and NES clustered together to form what the researchers identified as floor plate 2 cells. Meanwhile, the areas expressing MAPT and TH physically segregated themselves to form floor plate 1, which is the neighborhood where those crucial dopaminergic neurons live.
Yes. And the implication here is profound. Even outside the human body, lacking a centralized nervous system, lacking a vascular network to guide them, these cells possess an intrinsic driven ability to sort themselves into correct anatomical structures, and they do it based purely on local genetic communication.
That is just incredible. And I know it's a lot of biological acronyms for you listening. So to keep those neighborhoods straight in your head, think of the gene SHH as go to sleep on the floor, specifically floor plate 2.
I love that.
Yeah. And for the second neighborhood, think of MAPT as literally mapping the way to dopamine.
That's a great mnemonic. And, that spatial mapping proves the cells know where they belong, but the researchers, they wanted to go deeper than just observing the neighborhoods. They wanted to find the exact genetic levers that push a cell into one neighborhood versus the other.
Right. If MERScope shows us the map, how do we find the master switches that actually control the zoning laws?
Well, that requires analyzing gene regulatory networks, or GRNs. By synthesizing the RNA and the open chromatin data from earlier, the scientists identified 393 distinct transcription factors.
Transcription factors.
Right. These are specialized proteins that bind to specific DNA sequences. They control the rate of transcription of genetic information from DNA to messenger RNA. Basically, they are the ultimate master switches of cell fate.
Okay, let's go back to our factory analogy. They are the foreman on the factory floor, walking around and telling the workers which specific unlocked blueprints to pull from the manager's office and execute.
Yes.
But just identifying 393 foremen doesn't tell you what each one specifically does. Right? To figure that out, the researchers basically had to fire them one by one to see what broke in the assembly line.
Exactly. And to do that, they executed a multiplexed CRISPR-Cas9 knockout experiment to create mosaic organoids.
Okay, let's unpack that. Mosaic meaning what?
So, rather than turning off a gene in every single cell of an organoid, which would probably just ruin the whole thing, they took a pool of progenitor cells and used CRISPR to delete 12 specific transcription factors.
Just a select few.
Right. Then they mixed these edited cells back in with unedited normal wild-type cells and grew them all together in the exact same chemical bath.
Growing them together in a mosaic is brilliant because it controls for the environment, doesn't it?
Exactly.
Like if an edited cell develops differently than the normal cell sitting right next to it, you know with absolute certainty it's because of that missing transcription factor, not because of a weird dose of chemicals in the dish.
Yes. And the results of this mosaic experiment were striking, particularly regarding a transcription factor called OTX2. In neurobiology, OTX2 is heavily documented. We know it as a fundamental master switch required for midbrain development.
Right. And the mosaic organoids showed that when OTX2 was knocked out, those specific edited cells completely abandoned their midbrain destiny. Like by day 70 of development, they had shifted entirely to become hindbrain cells. So OTX2 is basically the steering wheel keeping the cell in the midbrain lane. You remove it, and the cell aggressively swerves into the hindbrain lane.
But here's the catch. If OTX2 is fundamentally required to build the midbrain, if it's the core engine of that developmental process, why doesn't removing it just kill the cell?
Yeah, because if you pull the steering wheel off a car at 60 miles an hour, you don't neatly merge into the next lane, you crash. Why does the cell just seamlessly transition to executing a completely different set of biological instructions instead of undergoing apoptosis and just dying?
Because cellular development operates more like a railroad network than an automobile. The researchers used the OTX2 knockout to demonstrate a really crucial mechanism called cross-repression.
Cross-repression.
Yeah. In the developing brain, OTX2 doesn't just drive the midbrain program. It also actively binds to and represses a different gene called GBX2. And GBX2 is the master driver for the hindbrain program. And here's the kicker. GBX2 operates the exact same way in reverse. It actively suppresses OTX2.
Oh, wow. So they are locked in this molecular tug-of-war.
Exactly. So when CRISPR deletes OTX2 from the genome, you haven't destroyed the cell's engine. You've simply removed the brakes from the GBX2 program. The second OTX2 is gone, GBX2 expression surges, the railroad switch flips, and the cell smoothly diverts down the hindbrain track, utilizing all those open chromatin blueprints we talked about.
Man, that cross-repression mechanism is incredibly elegant. It's just a binary switch.
Yeah, nature is pretty efficient.
So just to do another quick recap for everyone listening, transcription factors are the master switches of cell fate, right? And CRISPR was used to create these mosaic organoids missing certain TFs. Knocking out OTX2 shifts cells smoothly from midbrain to hindbrain. So think about this. If human development relies on these delicate binary genetic switches, how resilient do you think our developmental pathways really are to environmental changes?
It's a huge question in the field.
Right. But if CRISPR shows us what happens when we intentionally break the switch, is there a way to control it without destroying the genes? Like, can we just tell the cell what to do chemically?
Well, that transitions the study from observation to active engineering. The researchers conducted a massive comprehensive morphogen screen.
What exactly is a morphogen?
Morphogens are signaling molecules that govern the pattern of tissue development. They emanate from a source, and they form a concentration gradient. So a cell's fate is dictated by the specific concentration of the morphogen it's exposed to.
Yeah.
The scientists tested 48 different combinations of 10 different morphogen signals with names like retinoic acid, BMP4, SHH, and FGFs.
Oh, so they essentially played master chemists, just bathing different pools of cells in these 48 distinct cocktails.
Yeah, and to make sense of the results, they used computational algorithms to perform what's called trajectory inference.
Trajectory inference. Break that down for us.
Trajectory inference basically reconstructs a cell's developmental lineage. By analyzing the really subtle variations in RNA expression across thousands of individual cells, the algorithm plots the cells along a chronological pseudotime.
Pseudotime.
Yeah, it draws a highly detailed family tree showing exactly how a specific combination of morphogens forces a progenitor cell to branch off into a specific developmental path.
That is amazing.
Yeah.
Let's actually look at some of the specific recipes this algorithm uncovered, because they basically wrote a cookbook for the human brain here. Take retinoic acid, or RA. When they added RA to the dish, the trajectory inference showed the cells being driven toward becoming ventral medulla tissue, specifically glycinergic neurons.
Right.
And the multi-omic data explains the exact mechanics of how RA does this. It reaches into the genome and activates the transcription factors PX6 and PX7 while simultaneously repressing SOX6.
Exactly. Alternatively, when they applied a different morphogen called BMP4, the algorithm showed the cells branching off to become dopaminergic neurons.
Okay, so RA makes medulla, BMP4 makes dopaminergic.
Right, but the real power of this approach is in the combinations. If you mix retinoic acid and BMP4 together, the signals don't just cancel each other out. The cells integrate both inputs and form an entirely different, highly specific region, the dorsal medulla.
Wow. So they've mapped the blueprints, they've found the genetic switches, and they've perfected the chemical recipes to control those switches.
They really did.
But the ultimate question is, what is the clinical application here? Why do we want to generate these highly specific brain regions on demand?
Because the ability to generate rare, specific brain cells in a lab setting is a total paradigm shift for studying human disease. Take those glycinergic neurons they engineered using retinoic acid.
Okay.
In humans, mutations in the GLRA1 and GLRB genes within these specific neurons cause hyperreflexia.
Hyperreflexia.
It's commonly known as startle disease. It's a severe neurological disorder characterized by exaggerated startle responses and muscle stiffness, primarily in infants.
Oh, wow. And previously, studying the cellular mechanics of something like that in living human tissue was virtually impossible, right? Like you can't just biopsy the medulla of a living infant.
Exactly. It's too dangerous, too inaccessible. But now researchers can grow those precise glycinergic neurons in a dish, introduce the GLRA1 mutation themselves, and test therapies directly on the affected tissue.
That is a game changer.
It really is. And they also identified the morphogen recipes necessary to generate cerebellar glutamatergic neurons. These cells are deeply implicated in the pathology of autism spectrum disorders.
Oh, really?
Yes. So by generating these specific organoids, researchers can study how subtle genetic variations affect the synaptic development of these neurons in a totally controlled environment. Furthermore, the posterior brain organoids they've engineered are ideal models for studying pediatric brain tumors.
Because they can grow the specific region the tumor originates in.
Exactly. We can now cultivate organoids that express cancer-driving genetic mutations like ACVR1, BRAF, and NF1, and then systematically screen thousands of oncological drugs to see what effectively halts tumor growth in actual human tissue.
The potential for drug discovery there is just massive. But, okay, let's look at this critically for a second. The researchers are still ultimately dumping synthetic chemical cocktails onto clusters of stem cells in an incubator.
True.
Does this methodology genuinely replicate the intensely regulated, meticulously timed biological cascades of a natural human embryo?
Well, authors are actually remarkably transparent about the limitations of their model. You're right, a laboratory incubator is profoundly different from a human womb. These organoids lack a vascular system to deliver nutrients dynamically. They lack microglia and an immune system to prune synapses.
Right.
And as we discussed with the accidental hindbrain cells earlier, the chemical gradients applied by a pipette are blunt and uniform, whereas a developing embryo utilizes highly localized microscopic diffusion.
So the environmental context is just completely artificial.
It is. However, the trajectory inference and the CRISPR data prove a larger point. The intrinsic genetic programs embedded within the cells are astonishingly robust. Even with blunt chemical tools and an artificial environment, the cells correctly identify the molecular signals, flip the appropriate binary switches like OTX2 and GBX2, and execute complex developmental cascades with remarkable fidelity.
Right, they still know what to do.
Exactly. The model isn't a perfect replica of a human brain, no. But it is an unprecedentedly accurate simulation of specific neural regions that have historically just been completely inaccessible to molecular science.
That is just incredible. So to recap this final section for everyone, morphogens are chemicals that dictate cellular destiny, right?
Yeah.
And testing 48 combinations revealed recipes for rare brain cells. Retinoic acid makes medulla cells, BMP4 makes dopaminergic cells, and these organoids are basically the new frontier for studying things like autism, hyperplexia, and brain tumors. So if you're listening, think about this. Now that we have the recipe book for specific brain regions, what disease would you want researchers to model in a dish first? Because the possibilities seem kind of endless now.
They really are.
Well, we have unpacked a massive amount of biology today. We started by exploring how single-cell multi-omic atlases expose both the active manufacturing and the hidden architectural blueprints of developing cells.
Yep, the RNA and the open chromatin.
Right. And then we saw how Merscope spatial mapping revealed that even in a chaotic chemical bath, cells organize themselves into precise neighborhoods, like floor plate 1 and 2. We examined the molecular tug-of-war of cross-repression, where transcription factors like OTX2 and GBX2 act as binary railroad switches dictating cellular destiny.
The steering wheel and the brakes.
Exactly. And finally, we looked at how decoding morphogen gradients allows us to engineer specific brain regions to model devastating diseases like hyperplexia and pediatric brain tumors.
It is a profound demonstration of the underlying mechanical order of human biology. Throughout all the data in this paper, we've seen that cellular fate in the brain is intensely deterministic.
Deterministic, yeah.
It runs on rigid genetic programs, strict binary switches, and highly specific chemical concentration gradients. The development of our nervous system is, at a molecular level, almost algorithmic.
Like a computer program executing code.
Exactly. But I want to leave you with a thought. If the foundational building blocks of our brain are this rigidly programmed and deterministic, where does the chaotic, unpredictable complexity of human consciousness actually begin?
Oh, wow.
Right. Like, does our conscious experience emerge perfectly from the successful execution of this recipe? Or does the spark of humanity actually arise from the subtle, unpredictable errors and variations within that code?
That is an absolutely incredible thought to leave on. Thank you for joining us on this deep dive into the blueprint of the human brain. Keep questioning the instructions, keep looking for those hidden blueprints, and we will catch you on the next deep dive.